Okay, so we start again with a new session which is called Downstream Analysis. We will review some concepts of downstream analysis, or what we do after we have done all the statistical analysis. And the session will have two parts. One with the slides, and then there is also a small tutorial, just 2-page tutorial, so that you can do yourself some downstream analysis with online resources. Okay? So to start this session, I would like to start with one of the main databases that we use in proteomics, which is Uniprot Database. So I guess that some of you already know it, but in any case, I would like to stress the amount, or the huge amount of information that we have in this database for each of the protein entries. So for example, now in this particular case, I selected P53, and this is the page that we get from Uniprot. And in this database, so we first get, for example, so all the names, so gene names, different gene names that this protein has. We also have subcellular location, right? So we know, and there are the references linked in that define or describe the subcellular location of this protein. Also, not only for the protein itself, but for each of the isoforms that this protein can have, and this happens for P53, but with also for many other proteins, especially in human mouse, that are very well annotated, but also for many other species. So in here there are also expression levels in different tissues. There are links that links this entry to actually expression databases, so external resources that one can use once you have your candidates of interest, or your protein of interest. You can go here and actually do crosstalk with other databases to gather more information that has been already described in the literature to get most of your proteomics data. So other things that are in here are pathology descriptions. So what is this protein? In this particular P53 is a key protein in many cancer processes, but there is a lot of information for many other proteins related to what there are the processes or pathologies in which they are involved. There is also Go enrichment annotation, so in which biological processes, molecular functions, and cellular components this protein is associated with. There is also, and this is becoming more and more interesting, so these are the amino acid modifications that have been associated to this protein, so natural mutations that are or have been sequenced in either, in real samples, and then so both, so natural variants and also point mutations that some people in some papers have tried, and then there is also a brief description of what this mutation induces in this protein, and also always the reference to the original paper. There are also a lot of information about known interactors, and also again external links to other databases that are only interactome-based databases, right? And there are also, as we will see later, so graphs where you can see your protein interacting with other proteins, but all the primary information is also linked here in the Uniprot database, and from here you can navigate to many other resources. So this allows me to stress, right, the importance of Uniprot as a reference portal or reference web page to go and actually start browsing for your proteins. So there are also sequence information, again, not only for the canonical sequence, but also split in different isoforms. So here isoform 1, 2, I didn't put, but there are also the other isoforms for this protein. We also have phylogenetic links to phylogenomic database, to domain database, and also to many other like PTM databases here. And so every of these links to external resource that describes much better, for example, protein domains in different ways, right? And also, for example, structure. So what is downstream functional analysis? So downstream functional analysis is type of analysis that we do after we did our statistical work, and for example, we take all the proteins that are significantly changed, and we ask what the whole, not protein by protein, but all the proteins tell us, right? Assuming that actually the whole is more than the sum of its parts, right? And we will see now some, or we have already seen in the previous session, some approaches, for example, for class discovery. So for example, profile plot, heat map plot, and hierarchical clustering, PCA analysis, and also k-means analysis, right? And we saw this in the previous session. We will practice this afternoon. In general, what many times we are interested in is that actually we identify many hits or many proteins. We also quantify many proteins, and many proteins of the ones that we quantified result in significant fold changes in certain pathways, right? And then we deal with this list of 500 proteins, and the next question is, well, what should I do with this? Because I don't want, or I cannot go one by one and to Uniprot and check for each of them their particular significance in biology. And also this would not be quite advisable. And then the thing is, well, let's do some kind of analysis that actually let us go from this big list to a more reduced list that actually allows us to go into the more relevant proteins or the more relevant network nodes, right? And one way that I introduced already on Monday is actually, and that we will see more tomorrow with Martí Bernardo, is to do network models and actually see whether the proteins that are significantly enriched or changing in abundance are actually very close, interacting either functional or physically in a protein complex or a signaling pathway or a metabolic pathway, so that actually we can focus on the study of that particular function instead of a particular protein. Okay? So one way to do this, and probably also, or at least to approach, a first approach to this, is do Go enrichment analysis. And one of the web servers in which we can do this is this David functional web server. Here there is the URL where you can actually paste your protein identifiers and do a Go enrichment analysis. And what is a Go analysis? So Go are terms, right, that are hierarchically structured. So for example, the first term could be cell. So protein is in the cell. Okay, you have this tag. Then there are two other tags that are called, for example, cytoplasm and membrane, or nuclear and cytoplasm. And then within the cytoplasm, whether it's mitochondrial or peroxisomal or cytoplasm itself, right? And this creates a structure, and the question that we ask is whether within our significant proteins there is any of these tags which is enriched in our proteins that have a significant p-value. And also this concept of enrichment is very important, because what we have is actually, whenever we have a protein or a gene list, we will have these Go terms, right? And this would be the normal. So if we have 80%, so in the proteome we have 80% of the proteins that are cytoplasmatic. So here we will have 80% of the tags that are cytoplasm, okay? And then what we ask is, well, within my subset of proteins regulated, do we see more than this 80%, which would be the normal percentage that I would see, right? And again, right, so for example, I could have kinases, right? I know that, for example, 5% of my proteins in a proteome, it's kinase type of protein. Then what I would ask is, well, are within the proteins that are regulated, right? So I just take this subset and I ask, is this percentage 5 as I would expect, or is much bigger? It's 90%, right? This would mean that whatever perturbation I'm inducing, it's affecting very much all the kinome, okay? And then this would give me clues on the mechanism or the response that the cell is doing once we stimulated it with a particular stimuli. So what you would see, for example, this is one representation of Go term enrichment, in which you can see this hierarchical structure. the more red, the more significant, the more white, the less significant, right? So what we ask is, well, we know it has this tree structure, and then we have several tags that are indeed enriched, right? And then this can give us a clue of what is our perturbation or our treatment doing in the cell. So probably you are more familiar maybe with this type of representation of Go enrichment, which is actually a list of Go enrichment terms and a p-value, okay? And saying that, well, so my significant proteins are related to all this process with a p-value of less than 0.05, for instance, okay? But this should not let you forget that actually these Go terms are hierarchically structured and that there is a lot of redundancy within these tags, right? So here you can have, let's say, well, so there are a lot of things that are similar but more detailed because they are further detailed whenever we advance in this hierarchical structure. So another thing is how these proteins that are significant and that we have been interested in interact with each other. And as I said, so this can be physical interactions or functional interactions. And one of the one of the ways that we can do this is actually checking for these protein-protein interaction databases and ask for my given proteins which are their interactors. Okay? And then the second part would be to know, well, in which, for example, here, these proteins are colored according to their pathways. Okay? In which pathways are these proteins related to? And the question would be, I see several proteins that are regulated, and the question would be, in which pathways are they involved? And for this, we could use, and also many of you might know this, so the pathway enrichment analysis, we could use KEGG, for instance. This is a repository or a database that links every protein in the proteome to a particular function or particular metabolic or signaling pathway. And then we can ask, among my significant proteins, which are the pathways that are more enriched in this subset, right? And then see that, for instance, so for example, in this case, right, so that I would see that all the red or all the green ones are significant, so are proteins that are mapping to a certain pathway here or here, and I would say, well, so to see so many proteins for this pathway is not by random, but actually this is happening because this is the process that I'm affecting with my particular treatment. So here the green and red means the fold change, and you can see a particular pathway, how many proteins we have actually identified and quantified with significantly changes in abundance. To retrieve protein-protein interactions, and this is also part of the exercises that we have today, so we can use one of the best databases for this from EMBL, which is the string database, right? So here again you have the URL, and then we can put here our favorite proteins or our favorite 10 proteins or 20 proteins, or for example all the proteins that have been significantly changing from one group to another, and say, well, are they related somehow? So are they interacting within them? And then this will give us this type of graphical interface. And for example here, imagine, so the input that would be, for example, these 10 proteins that had a significant p-value, I put them here, and this is the graph I retrieve from this analysis. And what it says to me is, well, you know, it's not casual that all these proteins were significantly altered, because actually they are related to each other either by protein-protein physical interactions or by protein-protein functional interactions. And this can actually give me hints on what processes are going on with my treatment. Okay, so you can actually, so this database allows also to download all the protein-protein interaction data, so that you can also then mine it or do your own scripts to generate these plots if you don't want to do it through the web server. And finally, the last thing that I would like to highlight in this type of analysis is the motif enrichment. So we've seen the Go term enrichment, the pathway enrichment, protein-protein interactions, and then this would be the motif enrichment analysis. Here, this is meant for phosphoproteomic analysis, and again, so I induce, for example, or I inhibit or I activate a kinase, and then I check the phosphopeptides. And my question would be, all the phosphopeptides that are changing, do they have any motif in common? And if yes, can I associate this motif to particular kinase activity? And then what I would say is that actually my treatment is affecting the activity of kinase 1, 2, and 3. Okay? And I can do this with, for example, the website that I put here, which is MotifX, although there are also other ways to do this type of analysis. And this is very similar to, for example, sequence enrichment analysis of transcription factors. And finally, let me introduce the part of visualization of proteomics data that we will deal with it tomorrow during Marty's talk and Marty's tutorial, which is how we put all this data that we have, all this list of hundreds or thousands of proteins in a way that we can easily visualize them, easily say, oh, all these proteins that were significant are associated to this particular function, these other are related to another particular function, and these other are related to a third function, right? And we use a software that is called Cytoscape that actually allow us to define these protein-protein interactions, or retrieve them from KEGG or from known repositories. So it's not that we need to define how proteins relate to each other, but actually we can retrieve all this information from database, and then we can color them according to the changes that we have seen in our experiment, right? Either up, down, or just without changes. And so this you can see here, right? So this software allows us to have all colors and all type of colors for all type of experiments, and we can also color them in different ways depending whether it was in condition 1, condition 2, or condition 3, right? And it's quite flexible. We will see a lot of this tomorrow. You will do actually this kind of analysis with Cytoscape. And then what we can do actually is also isolate subnetworks. Say, well, you know, this is I had a big network of 1,000 proteins, but actually this batch of 15 proteins are changing, most of them, like here all in green or in red. They are highly connected among them. So this is a functional model that is altered during my treatment, right? And then I can make a nice figure about this and discuss why this model is highly regulated and in which functions it's involved. Okay? So one step further on this is actually start using Cytoscape and these networks to do prediction mathematical models, right? So what we can do is, for example, Boolean networks saying, well, this protein activates this one, this other protein activates this one, then what would happen if this would, right? If I perturb the cell in a certain way, right? And there are several ways of doing this. One is Boolean networks that we will see. So all the tutorial that we will do tomorrow, it's based on Boolean network modeling, right? And this allows us to predict phosphorylation responses of cells. Other ways to do this is with differential equation models that we will not enter into this in this course, but so that you know that there are different ways or mathematical ways to approach the modeling part, and that what allows us is that with the proper data from proteomics, actually train the model so that the model is predictive. And before doing an experiment, we can say, well, what would happen if I would inhibit this kinase? And then we predict this with the model, we do the actual experiment, and we then validate what the model said. And then we are confident that this model is working, and we can predict many other conditions. So with this, I would like to close this small introduction to this session, and then I would like, or you can start with the tutorial.